So in this lesson, now we're going to look at how to make biological drawing. So what are the principles or the rules of making a biological drawing? So biological drawing got two types, plan drawing and detailed drawing. Today, we are only going to do or learn about detailed drawing. So this is a microscope diagram with empty spaces in your notes, which you have to fill up so that you will know what is the name for different parts of the microscope. So this is your homework for this week. Okay. So when you make biological drawing, you must use HB pencil. So therefore, you need to check, do you have a HB pencil or not? Even if you're using mechanical pencil, make sure that you got a HB lead. Okay, it must be HB pencil. Okay, so if you don't have, please go and buy it either in the shop or buy it online. So that when we go back and do practical, you must have HB pencil. I will not allow you to make any drawings if you don't come into the lab without any HB pencil. I will not allow you to enter the lab because you've got plenty of time to get ready to have a HB pencil. And not only that, it must be sharp. Okay, so when you use your pencil to make biological drawing, your pencil must be sharp so that your line must be very fine. And then it is stated in mark scheme that it must be less than 1 mm in thickness. So if the thickness of your pencil line is more than 1 mm, minus 1 mark in exam. Minus 1 mark. Okay, that means you lose the mark. And your line must be clean single line. So that means you cannot draw a line and then you stop, you look at the slide and then you continue drawing, suddenly you got additional line here. And then you stop and at 1 line here. So it must be clean single line. And it must be clear. Clear means to say that I can see the line very clearly. Because sometimes the student, the line is very dark and then suddenly it becomes very fine. Okay, then after that, very dark again. So cannot, must be same thickness throughout. Okay, so this is called suddenly dark, suddenly not, suddenly light, suddenly dark, suddenly thick, suddenly thin, suddenly thick, cannot. Even thickness and same darkness throughout. Okay, and no feathery line and no gaps. So here, this is called feathery line. And then the gaps there. Usually when students draw circle, they draw like that one. What is this thing here? You can't see this in the actual specimen. So how come suddenly got one flagella out of the vesicle? So it's a new discovery. Suddenly there's a flagella growing in the vesicle. So cannot. So it's wrong drawing. And sometimes you draw like this. There's a gap here. Cannot. Circle is a circle. It must be full circle. Okay. Why? Because biological drawing means you are making a report. You observe the slide and you're trying to make a drawing to show people. So people were based on drawing to know, oh, okay, this is the structure of nucleus, mitochondria, and so on. So therefore, your drawing is a representation of the actual specimen. So you must report it accurately. So that's why your drawing must be accurate. Okay. So no shading. So in a biological drawing, we do not allow any form of shading. Either you shade the whole thing black color or use line, pattern, dots, everything. Don't have, cannot. Because if you go to actual specimen, you don't see this kind of pattern in the cell, okay, in the specimen. So never ever shade. I don't care what kind of shading, cannot. Okay. Now here, there are 4 diagrams. Which one of them is the biological drawing? So I give you a micrograph on the left side here, A. Anybody can draw this, right? An artist can draw it very nicely, but the artist actually don't know what he's drawing, right? B is an artist drawing. So what is a biological drawing? So C is a biological drawing. You outline the biological structure that you have learned, like cell surface membrane, nuclear envelope, heterochromatin, euchromatin, nucleolus, rough ER, smooth ER, mitochondrion. So these are the structures that you have learned and you're able to see in the cells, so you have to draw them. So can you see, even though in the cell, the nucleolus is dark, but you don't shade the nucleus, you only outline the nucleus because no shading. On the right side is the so-called textbook drawing. You don't memorize from textbook or notes and then draw it from your memory. Biological drawing is a report. You have to draw it from the actual specimen that's given to you, either in the form of slide or in the form of micrograph. Okay, so biological drawing is C. So can you see that a good biological drawing, right, when the size of the drawing is made the same as the size of the micrograph, they superimpose exactly with one another, fully superimpose. So that's called a good, accurate biological drawing. So the shape, the size, the location of everything you can see there must be accurately represented in the drawing. Okay, so here is an example of a micrograph. So the student is asked to draw 3 adjacent cells. Adjacent means neighboring cells that's touching each other. So if you're asked to draw 3 adjacent cells, you cannot simply draw any 3 cells. You must first look at the micrograph and choose the best 3 cells. The best 3 cells means the 3 cells where the structure inside the cell is very clear so that you can see clearly and you can draw clearly. So therefore, in this case here, the drawing on the right side is drawn from the 3 cells. Can you see what are the 3 cells? These 3 cells. Can you see or not? The location of the nucleus, the shape of the nucleus, okay, the length of the cilia versus the height of the cell are all accurately represented in the drawing. And even in the cytoplasm here, can you see this part of the cytoplasm is darker? So therefore, you will draw 2 lines to represent the cytoplasm is actually darker, but you don't shade it. Okay, so this is called the biological drawing. So I'll come back to the title later on. So when you're given a space in the exam paper to do your drawing, you must plan out your drawing so that you draw the object in the center of the space given as big as possible. How big? At least 3/4 of the space given must be filled by the drawing. That is considered big enough. Okay, at least 3/4. And then you must leave space for you to write your labeling. And then a space for title if you're doing it in your own notes. Okay, in exam time, you don't have to write title. You don't have to write title for exam time. But when you are going to a normal lab and you want to draw anything, you have to write a title. Because then when you go back home, you look at the drawing, you have to refer to title to tell you what have you drawn. Without a title, you just label cell surface membrane and nucleus. You do not know what cell is this. It can be any cell. So you have to label it with, you have to write a title for all the drawing that you draw in the lab. Other than exam time, you don't have to. Okay, because exam time, you don't take back the paper, you don't refer to the paper anymore. Okay, so when you after drawing, you have to label. So when you label, everything must be in pencil. Drawing in pencil, label line in pencil, label in pencil, annotation in pencil, everything in pencil. So that when you make a mistake, you can erase it easily because there's no additional exam papers given to you. There's only one exam paper. So you make a mistake, you erase, you draw it again in that space given to you. Okay. So how to label? So you label the nucleus, the label line, best is use horizontal and vertical line only. Okay, and the line must point into the structure. You see there's a space here. I want to label this space, I must point into the space. So for the nucleus, I must point into the nucleus. Cytoplasm, I must point into the cytoplasm. Okay, other than cell surface membrane is only this line. So I must point to this line and not nowhere else. So point into the structure using horizontal and vertical line, and the line must not overlap. They must not cross each other like this one here. Okay, and usually I'll label from top to bottom. I'll label from outside to inside the cell. So this is very systematic. Okay, so nucleus. So here I label the nucleus, and in the bracket is annotation. Annotation means description. So the name of the structure is nucleus. Annotation means description. So describe a little bit about the nucleus that you can see, right? Describe. So prominent means very obvious, very big, easily visible, prominent. And then it's purple color. So when you describe, you can describe the shape, you can describe the location, you can describe the content, you can describe the size, okay? You can describe the color, you can describe the texture. So it's called annotation. So there's a difference between label and annotation. You may be asked to label, you may be asked to annotate. So if you write label and annotate together, then label in front, bracket, annotate behind. If you're only asked to annotate, then you just annotate without the label and without the bracket. So for example, I'm asked to annotate the nucleus without labeling, then I label it as peripheral. It means I'm saying that the heterochromatin are located at the site of the nucleus. So it's called peripheral, means at the site. So this is called annotation, not label. Okay. Okay, so how about the title? How do you write the title? Title must have got number. So this is a first drawing, so it's called figure number 1. So when you go to the lab, you may be drawing 5 drawings. So your first drawing is called figure 1, second is called figure 2, and so on. Okay, so what do you draw here? You draw 3 ciliated epithelial cells. So I write here 3, what type of cell? Ciliated epithelial cell. And these cells come from which animal, which organism? So this is rabbit. Rabbit got so many organs. So which organ of the rabbit? Oviduct. And then this specimen can be all sorts of section. So what kind of section? Transverse section, oblique section, longitudinal section. So what do you mean by transverse, longitudinal, and so all this is like this? If you got a carrot, right? You go to the kitchen and pull out a carrot, okay? So you got a carrot. Now, when you want to see the cells in the carrot, you must make a section, a very thin section out of the carrot, okay? Make a thin section out of the carrot. That means you must cut it 2 times. You must cut it 2 times to make a very, very thin section, so thin that the light can pass through and the cells don't overlap. Then you take this section and you put it onto your microscope slide. Okay, put this section onto the microscope slide. Okay, put a drop of water, cover it with a cover slip, then put it into the microscope, turn on the light, and you can see the cells. So this is called section. Every time you say section means you have to cut the specimen 2 times to form a very thin section. So you can section this way across the length of the carrot. So this is called the cross section or transverse section. So this is called the TS. TS. Transverse section, which is also cross section. Okay, so if you make a section across the length of the carrot, right, across the length of the carrot, this section is called the longitudinal section because it's along the length of the carrot. And of course, if you cut it this way, right, this way, right, you make a section across like this. So this is called the oblique section, OS. So therefore, you have to indicate what is the kind of section that you're seeing in your specimen. Because different section, the shape of the cell will look different. Okay. So labeling, I already said it must be using ruler to draw the line for the label line. It must be ruler. Use horizontal or vertical line. It must end inside the structure. And there must be no additional thing to the line. That means don't add arrow, don't add the dot. It's just a pure single line. Okay. And the lines should not cross each other. And if you annotate with the label, annotate in the bracket. So there must be a title, except for during the exam, no need title. So title means got figure number, name of the structure or the cell or the tissue organ, name of the organism where it comes from, the organ where it comes from, and the viewpoint, right? Transverse section, longitudinal section, or oblique section. Okay. So this is an example of a drawing with the label and the annotation. Everything is okay with the drawing except for there shouldn't be arrow and dot. Okay. Everything is okay except for there shouldn't be arrow and dot. Because I get it from a book, so the book got this thing. Okay. Now this is a sample of a student's drawing. Can you identify the mistake in this drawing or not? So type one mistake that you can identify in this drawing. So can all of you, each of you type one mistake that you can see in this drawing? So Adrian said there's a shading, so there shouldn't be shading. And there is, you see these 2 lines, these 2 label lines, they cross each other, so it makes the drawing becomes very messy. Yeah, the line cross, label line cross. And also, can you see this drawing cannot superimpose exactly with the actual specimen? That means this is not a good biological drawing. It doesn't actually represent the actual drawing, the actual specimen. And a lot of features you can see in the micrograph, they are not drawn here. So when you're asked to draw this section, you have to draw everything about this section. So the student didn't draw everything. Okay, so, and then the size of the drawing is very small. You must occupy at least 50 to 3/4 of the size. Then the object is big enough so the detail can be seen. So a lot of mistake in this drawing. So you have got this homework in your notes where you got this drawing with empty spaces where you need to list the mistake in this drawing. Okay, so that will be your homework for today's session. And all these are in your notes as well to show you that in transverse section and longitudinal section, right, the structure will look different. So when you look at these two sections, you can actually guess what will be the three-dimensional shape of the actual specimen, because all specimens are 3D. But when you have to see in the microscope, you cannot put the whole carrot there. You have to section it. So when you section it, becomes 2D already. So how from 2D you can guess what will be the 3D structure, right? Like this one is cylindrical or rod shape, and this one is cigar shape, and so on. So please read this on your own, okay? So we have actually covered detailed drawings today. Everything you need to know about detailed drawings, I've already covered, all right? So any other details that I want to show you here? Yes, I want to show you a few more only, a few more. Now, a few more minutes, all right? So this is an exam question where students are asked to draw 3 epidermal cells and the adjacent palisade mesophyll cells below it. So 3 cells are drawn with 3 adjacent epidermal cells, and all, can you see, all the chloroplasts that are visible must be drawn. And then between neighboring cell wall, there must be a middle lamella. Okay, between neighboring cell wall, there are middle lamellas. And then the student is asked to label 1 palisade cell. So in exam time, you don't label everything. You label what the question asks you to label. So label palisade mesophyll cell, finish. Okay, so in detailed drawing, every structure that produced by the cell you have to draw. So you need to draw cuticle. In detailed drawing, everything that's produced by the cell inside and outside the cell must be drawn. So this is called detailed drawing. You draw the cell and everything that the cell produces that you can identify. Okay, and you choose the best cell that you can see to draw. Now this one are 3 drawings. Can you see the mistake in the drawing? A is very accurate. B got shading, so it's wrong. C is not accurately representing because you see not, there are so many chromosomes here, but suddenly only 4 chromosomes. So this drawing is not accurate already, right? Now here, how do you label? You have to point into the cell, but this is wrong because you are pointing to 1 cell, okay? But how come there's an S here? So it's wrong. This one also is wrong because you need to point into the cell. This one is correct, okay? This is wrong because you have to point to the structure. This is wrong because if you use bracket, the bracket must touch the cell, or else you are bracketing, you're putting the bracket to the air and you're labeling the air gut cells wrong. So you must touch the cell, the bracket. So this is correct. And this is wrong because it's pointing to 2 cells, there must be an S here. So singular and plural applies into the labeling. So again, these are 3 samples of students' drawing. All of them are quite good, but actually only 1 is correct. Only this one is correct, even though it's not accurate, because you see not, this one got middle lamella. The rest of them no middle lamella. So even though the rest of them are quite accurate, I can see that A and B are more accurate than C, but C score full marks. A and B don't score full marks because no middle lamella. Okay, so that's all for today. You have learned the differences between prokaryote and eukaryote, and also similarity. You have learned about the virus structure, and today you have also learned about how to make a biological drawing. Okay, so the homework that you need to do today, okay, is this one here. Can you see or not? Am I sharing? Share screen. So this one here is a homework that you need to you need to do. So this can be found in the exercise folder in your Dropbox. So in the exercise folder, right? So there's this exercise for detailed drawing where you need to refer to this micrograph and you only need to draw one cell. Okay, pay attention, you only need to draw one cell from the micrograph. So choose the best cell and draw as accurate as possible. Right, from the micrograph, only one cell, and label it fully. Okay, so this is a homework you will need to pass up. So later on, I will